Let's call it.
Welcome everyone back. We will have 4 talks. One of them is 30 minutes. The remaining 3 are short talks. The first one is Linda Kenney. She looks like going to talk about Sita Rama. No.
Yeah, definitely. It's Deepawali after all.
Stage is yours.
Oh, right. So we were talking last night about the Ramayana and whether Sita got a bum rap or not. So what do you think? As a feminist, I think, you know, she waits and waits and waits for Rama to show up. He finally shows up and then he won't have anything to do with her because maybe she's no longer chaste. I mean, that's just crappy, right? I think we need to write a new version. Anyway, so that's just my perspective. Anyway, it's really good to be back here in Bangalore. Mike and I came here many, many times, starting in 2010. We have lots of wonderful colleagues. It's great to see again after COVID. And I want to tell you about our work. And maybe I'm thinking this might even be the last scientific presentation I give on my work. I don't know. You never quite know when that is, but maybe this is it after our crazy president and the state of Texas have combined forces against me. This is now the view from my office, which is also from my home in Santa Fe, New Mexico. And I am now president of Mechanobiologics, and I will be telling you about that work, I guess, later this afternoon. But for now, I'll tell you about what we've been doing with Salmonella. And most of this work, at least, was started when I was in the Mechanobiology Institute at the National University of Singapore. And I think where we had this wonderful combination of interdisciplinary scientists, physicists, chemists, engineers, microfabricators, biologists, and me as the lone bacteriologist. So I want to tell you about what we did there. But first, let me just describe very briefly what happens during Salmonella infection. So we get an infection from ingesting contaminated food or water. Most of the bacteria gets killed by the extreme acid pH of the stomach, but the bacteria that survive go on to the intestine, and Salmonella, through its signal transduction systems, recognizes that it's in a host environment and turns on a virulence program that drives its uptake across the intestinal epithelium through what's called a type 3 secretory system that causes actin ruffling and membrane ruffling and promotes its uptake. And then once Salmonella is inside cells, it turns off that system and it recognizes that it's in a host through the acid pH of the vacuole. It's internalized into a vacuolar compartment, and then it turns on a different type 3 secretion system that we refer to as SPY2. It's encoded on a pathogenicity island that was acquired through horizontal gene transfer. So we're going to focus entirely on that pathogenicity island too. So the question is, how does Salmonella know when it's inside a host and it's time to turn on its virulence program? And this work started with Smarajit Chakraborty, who is a postdoc in Singapore. And we used the iSwitch, which was created by Yamana Krishnan when she was at NCBS. And I think I maybe found out about the iSwitch on that first visit to NCBS in 2010. So the iSwitch has 3 oligos, 1 here and 2 others, and there's a mismatch in the middle, and that enables it to act like a hinge. And at the ends, we have unpaired cytosines and 2 different fluorophores. So under acidic conditions, you get non-Watson-Crick base pairing of the cytosines, and the fluorophores come close together, and we can see that as by measuring fret. So Smarajit put the iSwitch inside Salmonella cells, and these bacteria now are, you see here, remain around neutral pH for the duration of the experiment. But if we take those cells and infect human macrophages, then what we see was an immediate acidification. So the pH drops quite dramatically here. And that acidification is dependent on the signaling system, the MZ-on-bar system. And so we knock out on-bar, the bacteria remain at neutral pH. They fail to turn on virulence. We can inhibit the vacuolar pH through bafilomycin and show that once again, the bacteria are no longer acidified and they're avirulent. Okay, so then Samarjit went on to ask what happens in cells when they've undergone this dramatic pH shift. And we looked at AMPR-dependent gene regulation under these conditions, and about 1/3 of the genome is changing. It's quite an extreme environment. And what Samarjit showed is that there's normally amino acid decarboxylation systems that restore neutrality. And one of them is the CAD-ABC system, where CAD-C is a regulator that's in the membrane. And there's a lysine decarboxylase CAD-A and a transporter CAD-B. So once the lysine is decarboxylated, it's transported out of the cell as cadaverine. But AMPR is keeping this system turned off, so the bacteria remain acidified. And then that drives the expression of another two-component system, SSRA and B, which is on the pathogenicity island. And SSRA and B regulate the transcription of the type 3 secretion system, as well as the virulence proteins, which we refer to as effectors that are secreted through this SPY2 needle complex. Okay. So the question that we were really focused on is how does acidification drive this activation of SPY2? So we know that the FOPQ system is involved in activating SSRA and B and MZ-AMPR as well. And then the SSRA-B system drives the transcription of this entire pathogenicity island. So we turned to the imaging facilities that we had in the Mechanobiology Institute, and there was a big revolution in imaging, as I'm sure you know, that we could go from images that look like this in 2013 to images a couple of years later using super resolution imaging, collaboration with Mike Heilman at Goethe University in Frankfurt. So we can image the bacterial nucleoid by EDU staining. And then these are AMPR molecules. And you can see that we can visualize individual molecules and we can count them, right? So and we can also stain the bacterial membrane with Nile red. So using these approaches then, we asked what happens under acidic conditions to the SSRA-B system. So we made photoactivatable fusion proteins, and we always test to make sure they're active. So for the regulator SSRB, the fusion is at the N terminus. For the kinase, it's at the C terminus. And you can see here in individual cells, then we count. And in every case, the SSRA kinase increases under acidic conditions, and the regulator goes up quite dramatically as a function of acid pH. So, and that's different from other two-component systems like the MZ-AMPR system, where the numbers remain relatively constant. So it's a unique feature of the SSRA-B that they're upregulated under acidic conditions. And now the problem with studying the SPY2 type 3 secretion system is that it's not abundant. We have really beautiful structures of SPY1 because there's like 80 copies per cell and they spew off. So people who are studying the flagellum, of which these systems are homologous, could even find them sort of in the, on their grids when they're looking at flagellar structure. But the problem with SPY2 is that you see it's not abundant and it's also phenotypically heterogeneous. So not all of the cells actually commit to making a SPY2 type 3 secretion system, even though they're all responding to the acid pH of the vacuole. And that makes it more difficult to study. So we have an antibody to the translocon, or I should say we had an antibody to the translocon. And you can see now that not all cells are making functional apparatuses here, and that you see about 1 copy per cell, and it's usually at the pole. All right. So then we wondered whether this heterogeneity was due to a heterogeneity of expression of the SSRA-B system. So we go back and ask, we can count molecules in the cells that are actively making the type 3 secretion system here. They're identified by these spots where the antibody is recognizing the SSEB. And we count those particular cells versus these that are not making an apparatus. And you see that there's really no difference in the number of SSRB molecules. But of course, what we don't know is it's likely the phosphorylated SSRB, which is important, and we don't have the ability to discern that here. But what I will tell you is now, after many years later, we have a better understanding of the pH of the vacuoles, thanks to the work by Kiran Singh, who was a postdoc in the lab, both in Singapore and in Texas, and he's now just returned to Manipur at a new university and as an assistant professor. But so Kiran made a mutant of M. cherry, and its fluorescent lifetime is pH sensitive. So we could use fluorescent lifetime imaging microscopy to measure the pH. And we did that first here in these cells that are clamped at various pH values. And so you can see that the lifetime of the probe increases as a function of pH, and it increases quite almost linearly right here. And this is in vitro in cells. And now, and we show that when we then put the M. cherry inside Salmonella cells and infect HeLa cells, then we are looking at intracellular bacteria. And you can see we're also staining with LAMP1 to determine which of those are in vacuoles. And what we found was really surprising. And that was that there's two different pH values that we see, an acid pH group and a neutral group. And we thought that the acid pH group would be the bacteria that are in vacuoles, and the neutral group would be out in the cytoplasm. But you can see here where the LAMP1 is surrounding these cells, but it's clearly a neutral. Now, what we cannot tell is whether maybe the vacuole is damaged in some way, but it's absolutely clear that we have two distributions, both at early and late infection times of pH. And so we would argue that only in this group where the bacteria are in acid vacuoles would be the group that are actually going to make a SPY2 type 3 secretion system. So we think that that heterogeneity that we saw is because of this vacuolar heterogeneity. And I should point out too that that work was just published in ACS Sensors in 2025. All right. So then we can also use our SSRB fusion protein to do single particle tracking and look and see how acid pH affects its DNA binding properties. And so when we do that, here are the tracks that we see at neutral pH compared to acid pH. And the diffusion coefficients that we measured are then color-coded here in this heat map. And so what we see is both nonspecific binding and specific binding increases quite dramatically. It doubles as we go from neutral pH to acid pH. So the level of SSRB goes up and its DNA binding increases under acidic conditions. So then we teamed up with Anjana, who's a physicist at the Mechanobiology Institute. We did a lot of work with him using single molecule approaches to study HNS properties. And with Ranjit Gulvadi, we used this single molecule unzipping assay, which we also used with HNS to study the binding properties of SSRB. By the traditional methods of measuring DNA binding, it's really tricky to understand how acid pH affects the binding, right? And so in this assay, we have a DNA that's tethered to a cover slip. Then it forms this double-stranded segment here, and it's tethered to a magnetic bead at the other end. And so when we pull on this, we can measure the unzipping force, what it takes to open up this DNA in the absence of protein. And then when we add SSRB at varying concentrations, then we can measure not only the change in force it's required to unzip this complex, but the extension that we see when it's unzipped. Right? So when we plot that then, we basically get a binding probability. And so you can see at neutral pH, we get a dissociation constant of around 150 nanomolar. And you see that it's highly cooperative at neutral pH and at acid pH. And what we see here is about a 30-fold change in binding affinity when under acidic conditions, right? So again, SSRB levels go up, it's more protein is bound to DNA, and its DNA binding affinity changes with acid. And so Dasvit Shetty was a graduate student in the lab and started in Singapore and then came to Texas. And so she wanted to understand what's the molecular basis for this acid sensitivity that we see with SSRB. So we had an NMR structure that we had solved back in 2009 of the DNA binding domain. And we had this construct here where we just cleave here at the linker region where we can separate these two domains. And so what we found is that the pH sensitivity, interestingly, was not in the DNA binding domain. It was in the receiver domain, which is the phosphorylation domain. All right. So then we went on to say that maybe likely residues that could contribute to this acid sensitivity would be histidines because their pKa is in that range where we're seeing these pH changes, right? And we also, Smarajit had measured the pH dependence of secretion, and he found that there was, the threshold was around 6.5. All right, so we wondered, are histidines in SSRB responsible for its pH sensitivity? So we have in the N-terminus, we have 4 histidines. And so 3 of these are non-conserved when we look at other members of the SSRB family of regulators. And one of them is highly conserved. So to be brief, Dasvit started out with each of these individually, and they had no impact on the acid sensitivity. And then she finally knocked out the conserved histidine. And what we see here, back to our unzipping assay, is that this histidine 12 substitution loses the acid dependence of DNA binding. So we get a KD that's very similar to the wild type at neutral pH. It's still cooperative, but you see we completely lose cooperativity, and the affinity is really no change compared to the neutral condition. So we have a single amino acid in the receiver domain that is affecting the acid-dependent binding of the C-terminal domain. So it's classic allosteric. When we put it inside Salmonella cells and then infect HeLa, you see this is the wild-type protein. We are looking at a reporter that is one of the genes that's driven by SSRB. It's highly acid-induced. And you see that the histidine substitutions have the same neutral activity, but they no longer undergo that acid-dependent increase, right? And then when we look at their ability to replicate, so you can see here's our wild type, they're clearly impaired in the number of bacteria that you see recovered from the host, and that's shown here, that basically it's as similar as a vector control. There's no acid-dependent increase in replication. So I should just emphasize that a single amino acid in the regulator is completely controlling virulence, because if we knock that out, it then does not undergo this pH shift, right? So then we are interested in trying to image the effectors that are secreted through this Phi2 type 3 secretion system and just understand their impact on the host, because they're modifying the vacuole so it's not degraded by the normal endocytic pathways, right? So then the Salmonella can replicate within this acid compartment. And so what we see in this super resolution SIM image, here's the bacteria in turquoise, here's the Salmonella containing vacuole, host nuclei here is stained with DAPI. And as a result of the Salmonella infection, you see these what are called Salmonella-induced filaments or endosomal tubulation. So these virulence proteins of Salmonella are taking over the host trafficking machinery and resulting in this endosomal tubulation. We were hoping we could sort of follow that in super resolution. So a graduate student particularly interested in these 2 effectors, Salmonella secreted effector J and Salmonella induced filament A. They have a sort of yin and yang effect on one another, and they interact with a kinesin interacting protein, and then they affect microtubule motors, kinesin directly. So, but the problem is that when we try to image photoactivatable fusion proteins, that they're jammed, they don't get secreted. So we then got around this problem by making HA tags that were then secreted, and then we could use immunofluorescence to follow the effectors in the cytoplasm. Okay, so when we do that, and this is SSEJ after a 16-hour infection, we see that it's decorating these salmonella-induced filaments all along the filament here, right? And here's the sort of salmonella-containing vacuole. It's emanating off of the vacuole. But unfortunately, wherever we put HA tags in CIF-A, we could not, it was not effective. We did this N-terminus to C-terminus. We had a crystal structure. We put them in intracellular loops. Everywhere we worked, it basically didn't work. So then we turned to genetic code expansion. Again, Kiran was an extremely well-trained chemist from Soban Chen's lab in Delhi. And so he is looking to visualize this CIF-A by genetic code expansion. So what we do in this case is we introduce a mutation in the CIF-A gene, and it is going to encode an unnatural amino acid at a particular site. He tried several different sites in the beginning, so we were guessing, but several of them worked. And then we express that in a background where we provide the orthogonal synthetase and the tRNA that's going to recognize that codon, right? And then deliver the unnatural amino acid, which then is secreted by the type 3 secretion system. And then we can site-specifically label using click chemistry at that site. And so then what he shows is that we can now label this CIF-A protein and visualize it. In the absence of the unnatural amino acid, you see that there is virtually no background here. We could also use fluorescently labeled unnatural amino acids, of which now there's a whole repertoire of new probes that are available. And there's a wonderful website for this from the guys at Oregon State University in Corvallis. So we could use these and avoid the click reaction processing. So we can visualize CIF-A. And I want to show you how this GCE labeling is superior to immunofluorescence. So in this case, we're labeling SSEJ. It's got an HA tag, and it's also got a mutation that encodes for the unnatural amino acid. So we can do both kinds of labeling and directly compare the same protein. And what you see here is when we label through genetic code expansion, we get this level of CIF-A labeling. Here's what we get from immunofluorescence with the HA tag. And you can clearly see that there's a lot of background that's present when we use immunofluorescence that we lose by genetic code expansion. So it's a really beautiful way of labeling. And we were also able to label proteins that were very low abundance. And one of these is SSAP. It's believed to be the gate that's involved in determining when the system switches from secreting the apparatus proteins to when it then secretes the effectors. And when we made a fusion protein of that, it was cleaved. So it didn't tolerate anything, but we could then site-specifically label it and show that at late time points, it's actually secreted. And finally, I can show you that we also used it to label a Zika coat protein. This was a precursor experiment for what we hoped would be a COVID viral labeling, but we never got around to that. But anyway, we could label Zika virus coat protein and produce fluorescent virus, which we could then use to infect naive cells and watch viral uptake by a loss of fluorescence. Again, really powerful method. Okay, so what I told you about here is SSRB regulation of SPY2, which gets turned on by the acid pH of the vacuole. I didn't talk about this, but SSRB is de-repressing HNS on the pathogenicity island, as well as activating transcription. But what we also showed that SSRB was important in driving the carrier state, which we see is driven by biofilm formation. And that happens under neutral conditions where the kinase is almost absent. SSRBD represses HNS at the CSGD promoter, that's the master regulator of biofilms, and it turns on biofilms. And I just want to show you that we were able to actually visualize biofilms in a heterologous host. So we could, and this was nucleated by a collaboration in the Mechanobiology Institute, where our neighbor is Ronan Zeidelbar, and our postdocs got together and basically came up with this experiment. So normally you feed C. elegans E. coli, so we can switch the food source and feed them Salmonella. And then when they're labeled, we can follow the Salmonella. And what was interesting in this system is that Salmonella formed, again, because I can go back to Kieran's pH probe and show you that we could then measure the pH. Here's a heat map of the height of the biofilm. So it goes up to 25 microns. And we could then take different Z-stacks and measure the pH across the biofilm, which in the case of Salmonella does not vary very much, from about 6.6 or so to around 6.9, just demonstrating the versatility of the probe. We could also then measure the pH of the inside the C. elegans intestine, right? So here we let them feed Salmonella, and you can see that the pH in the head is neutral, and that's where we see the biofilms form, and it becomes more acidic as we get closer to the tail. And finally, this one's for Tim. We could then use zebrafish as another model for studying Salmonella, and they're optically transparent. And what we see is that when we infect Salmonella, when we infect this, go to this particular region designed to take up proteins. When you just feed them cherry, you see a highly fluorescent region here. And now we can, when we see Salmonella infection, it was really hard to tell if they were just lining the lumen or if they were actually being actively taken up. But when we put the cherry probe inside Salmonella and then measured the pH in these different compartments, what we could see is that in the anterior of the fish, it's very neutral pH. And then in the LRE region where the Salmonella are, the pH drops considerably. So we would argue that they're being taken up and they're inside vacuoles in the endothelial cells of the fish. And then, so because we have these competing lifestyles of Salmonella that are both driven by the same regulator, the virulence lifestyle and the biofilm lifestyle, we were really hoping to try to understand how does genomic structure change with these lifestyles. And so what we have in super resolution is not a very informative picture of the nucleoid. It's really basically a blob. So how can we do better than that? And like we know under the virulence condition that this pathogenicity island is being opened up because a 32 kb piece of DNA is being activated transcriptionally. So the structure has to change. And can we visualize it? So we collaborated with the new assistant professor in the biochem department, that's Guy Neer. And so what we divided the Salmonella chromosome up into 20 steps of each about 250 kb. And we ordered site-specific oligonucleotides that bind in each of these different regions, and they are bar-coded so we can determine what region they come from. And we have hundreds to thousands of oligos in each of these various regions. And then we image, so we stick the cells down on cover slips, permeabilize them, and then we flow in oligos from step 1, and we image that. And then we flow out those oligos and flow in the next set, et cetera. And it takes 4 days for us to image one bacterial cell. And so what we have thus far is this picture. And each of these dots is an oligo that we imaged. And the colors are coded by the barcode. So we know exactly where this region of the chromosome is based on the barcoding. And so we have an entire genome. I believe it's the first at super resolution here. And what I was hoping is that we would be able to say, here's a prototype of a virulent state. Here's a prototype of what the chromosome structure looks like under biofilm state, right? But there is so much single cell variability, right? That thus far, we don't really have a good snapshot of what the neutral condition is, say. And I have to say that as we examine the data, it looks like, the chromosome interaction domains are really not very apparent at the single cell level thus far. But anyway, so that's an ongoing piece of work that we're hoping to, and now we can dial down on those particular regions, and do much more higher resolution of, say, the SPY2 region under acid pH, for example. But so what I told you is that the bacteria acidify in the vacuole, it's OMPAR dependent. We see an increase in the SSR-AB system, its affinity for DNA increases, its binding increases, and the basis for this acid sensitivity is histidine 12, and the challenges of studying SPY2. And now we can image these effectors using genetic code expansion. I didn't show you this work, but the whole tubulation formation is force dependent. And we can, again, visualize things that we couldn't do before. And so I will just leave you with that and with a precursor to talk about how we're going to use low frequency ultrasound, which I guess I'm speaking about later this afternoon, and some of our recent papers using low frequency ultrasound to treat age-related maladies. And thank you very much. Let me just go back to, my new life. And this is our Deepawali party back in Texas. And so here's Kieran, who did the GCE, as well as the pH probe, Dasvit Shetty, the acid dependence of SSRB. And thank you very much for your attention.
Thank you. Questions?
Thank you, Linda, for this nice talk. So I have a very nice question. So you say that SPY2 is expressed when you have a low pH, that's clear. But my question is more related, what triggers this change in pH? Are these cells the ones that are going to express SPY2? They express something that the change comes from the eukaryotic cell or from Salmonella in terms of pH? What triggers the change
okay.
of pH?
Well, so they respond to the environmental pH. So they can respond to the environmental pH in vitro. And then there are OMPAR-dependent pathways and OMPAR-independent pathways that enable them to survive an acidified state. And so many tRNA genes are changing under these conditions. They start to use nitrate as a carbon source. And that's one of Smarajit's very extensive papers where he analyzed, especially we were interested in the OMPAR dependence. But then there's numerous other pathways. There's other amino acid decarboxylation systems with different PKs that come into play depending on how acidified you're talking about. So it's an intrinsic property of both E. coli, Salmonella, many bacteria, and that makes them unique from eukaryotic cells, which tightly regulate their pH to be, in the same range of 7.35, 7.45.
And just one more question. Do you know if HNS DNA binding change with acid pH?
It's funny. So Ranji, I don't know. I don't think he looked at that. Yeah, he's set up the whole assay and everything, but we were more looking at particular amino acid residues that were important for binding. But like whether, okay, this would be a great experiment for you to do, right? And because, yeah, right. So you could absolutely, as de-repression easier at acid pH, right? Or even at neutral pH where we know SSRB binds at the CSGD promoter and destabilizes HNS binding. And then it doesn't even have to activate transcription just by driving off HNS. It enables the normal transcription pathways to occur. So understanding that in your system would be great.
Thank you.
Yeah, thank you.
Unsurprisingly, going to ask some imaging, some imaging questions. So first in the fish, like
Yes.
obviously it was quite low resolution imaging of the
Yeah,
structure.
that's the
So
problem.
are you able to actually dissect out tissue and look at it in high resolution to see
Oh.
where exactly it is?
Yeah, yes, we did. I have some images, but I should have put those in for you. I'm sorry. I didn't. Yeah, we did. We dissected out the intestine but then the student went to another lab. So it's like she got really good at it and then left. So we lost that. But we did do some early beautiful images there. Yeah.
And the second sort of question, so you mentioned earlier in the talk that you did kind of a test where you put the Salmonella into HeLa cells.
Yes.
Response. Were you able to maybe look at, say, a gut organoid, so I guess a more physiologically relevant system, and then see in the human how it would respond? Because HeLa, of course, are always very weird. So
Yeah, I know. Those would be great experiments to do, but no, we didn't do that.
Question here. So
Line
the
three.
pH stratification that you showed, function of depth. In the intestine, in the intestine, worm intestine, biofilms don't have a directionality, right? I mean, I'm just wondering, so growing them in culture, you have surface, with air on one side, water on the other side, I guess. In the gut, what governs that?
What governs the pH?
Yeah.
Oh, I'm really curious to see if any C. elegans people are gonna pick up on those measurements that we made to see, because that sort of surprised me that there was this gradient from the head to the tail, right? I don't know. I don't know anything about it. And like, why in the C. elegans intestine do the bacteria stay in the intestine? I mean, I think they start forming biofilms because the pH is conducive to that, but why aren't they taken up? And is it because the glycocalyx is kind of, providing an opposing force that's preventing uptake, or SPY1 doesn't get turned on? Or, there were all these questions that would have been interesting to answer that I don't know. Yeah. Madhan.
Hi, so maybe I missed it, but what is the endocytic route that gets the Salmonella into the host cell?
What is it? So their uptake is catalyzed through actin and through the impact of the virulence factors of the first type 3 secretion system. So that helps drive their uptake. And then they're in a vacuolar compartment, right? And then they basically, I believe, prevent lysosomal fusion through this endocytic, through their virulence factors, so that they don't get killed. And then they can replicate within this compartment, and then they disseminate in an unknown process.
The mechanism for the tubulation, the spontaneous tubulation.
Yeah, the tubulation, well, it's driven through effects of SSEJ, CYP, a whole bunch of the other factors, working through kinesin on microtubules, and right, so it's force driven through the action of kinesin. Yeah.
Last question. My question is same as Madan's, what drives the tubulation? Salmonella are motile bacteria, right? And
Are motile, yes, but
So-
okay, but as soon as they turn on the type 3 secretion systems, they repress flagellar expression. So they are sensing, and likewise when they're in a biofilm, the things that activate biofilm formation represses motility. And turning on the type 3 secretion represses motility.
So the tubulation, you think, is nothing to do with?
No, nothing to do with motility. Yeah, thank you though. All right, thank you very much.